believe it started here. All right, so hello everyone. For our presentation in the science and news, we decided to do our presentation over how FDA approves the CRISPR gene therapy for sickle cells. My name is Christian Williams.
I'm Daniel Gardner.
And I'm Dylan Babst.
So to start off, we're going to give you guys a little bit of an overview of kind of what we're going to be going over in our presentation today. First, we're going to start off with sickle cell anemia overview, kind of give you guys some background on what sickle cell is, what it kind of does to the body, and kind of how things have been corresponded within the last few years as far as the grounds in which they've been making into performing a cure to help solve sickle cell. Next, we're going to get into our article over the gene therapies of sickle cell. That's an article by CNN. Then we're going to go into our peer-reviewed, which we did over, it was a New England journal that kind of goes into that. And then following that, we'll get into our science communication and kind of how does it compare. So our comparison between the two. So what is sickle cell anemia? Sickle cell anemia is known as a disease. It starts with the red blood cells that become shaped like sickle cells or like a crescent moon. The cells are stiff and sticky. So with that, they can be stuck. They can get stuck in like small blood vessels that ultimately leads to the blocking of blood flow. They also break down faster than normal typical cells, leading to a shortage of red blood cells known as anemia. In order to get sickle cells, a person must inherit the faulty gene from both parents to have the disease, and it's usually most common in African Americans. And until recently, there had been no universal cure for the disease. On this next slide here, as you can see, we have what sickle cells look like through the bloodstream. Like I said before, it's kind of a crescent moon type of thing. The sickle cells, they block airflow. It's very painful for people who have this disease. On the right side, you can see body shape to where it shows the normal cells, what normal cells would look like, what sickle cells would look like, and then what acute sickle cells would look like.
So in recent years, there's been a new advancement called CRISPR gene therapies to help combat sickle cell disease. And CRISPR is a term that stands for clustered, regularly interspaced short palindromic repeats. And that's a mouthful. It's known as a genome editing tool that allows scientists to basically alter the DNA sequences and modify gene function. In scientific terms, it's called as a pair of molecular scissors. So there are two gene therapies called Casgevy and Lyfgenia. So Casgevy was the first FDA-approved CRISPR gene editing therapy for sickle cell anemia. And it uses patients' own blood cells. Their blood cells are collected outside the body, and they use a enzyme called Cas9 enzyme, which cuts the DNA at the targeted site. And going along that process, it pretty much then tries to repair the break during which scientists can insert or delete the genetic material. Going into this third slide, Lyfgenia is a lentiviral gene therapy that adds a functional adult-like hemoglobin gene into stem cells, kind of like the model before. Patient stem cells are genetically modified and cells are reinfused after chemotherapy, similar to Casgevy. And the impact of this has seen very positive results. You see right here as 28 of 32 participants, or 88%, have experienced complete resolution of vaso-occlusive events over 6 to 18 months. So in a short time, a lot of pain relief has been made for people suffering with sickle cell, and that's a big highlight of this new advancement. However, since there's not a lot of information about it, we don't know in the long term. And from what we found in our article, there are some cons or risks associated with it. It is a highly expensive treatment, which can cost you around 2 to 3 million. But however, they are implementing kind of a kind of to help with the payments and stuff. They only tie the payment to how effective the treatment is over time. There are risks of cancer as well as infertility, and potentially it could affect off-target sites by cutting DNA elsewhere.
Okay, so now we're just going to dive into basically the New England Journal and talk about the peer review article. And in this article, they basically report results from the clinical trials of CTX001, which is basically just a scientific name of basically talking about sickle cell, and it's given to the name, the brand name of Casgevy, which is just like the scientific name of it, and that's what they use to talk about Casgevy in this article. So CRISPR in this article is used to edit BCL11A gene in the stem cells, as well as the goal of this is to increase the fetal hemoglobin to stop sickling, which, as Christian kind of gave you background on, that's just the shape of red blood cells and how that causes severe pain to individuals who have this disease. And then they kind of give more detailed data on the HbF levels, which is just another short term of the fetal hemoglobin levels. And it also talks about the side effects and the number of patients that they have helped, as well as many patients that were used in the trial. The difference between this article and kind of the article that we use from CNN is that this article had many cited sources, tons of stats and charts that kind of were better suited for someone who is a scientist or something like that, or a doctor using this article for reference or trying to read this. They use a lot of technical terms that kind of make it to where someone who is not like me or you is reading it and understands exactly what they're talking about and exactly what they're doing to ensure that people who have this disease are able to have care and get rid of it. So as we go into the comparison, I'm just talking about both of them. Both articles provided a lot of valuable information on the two, the FDA-approved gene therapy of Casgevy and of Lifagina. The article also focused on tools that would be needed and the financial aspects of how much does gene therapy cost. As Daniel stated, it's crazy. Just between both of these two diseases, treatments are between over $5 million, not 5,000, $5 million, which just is kind of absurd to even think about and kind of wrap your head around how expensive these treatments are and how much money is going into it to ensure that we get rid of this disease. The peer-reviewed article shows us the results of the various clinical trials, and unlike the CNN article, it doesn't do that. It doesn't really go in depth to that. It just kind of talks about the viewpoint of the patient and their well-being, as the peer review article goes way more into depth, talking about like how they actually, this whole process works and how they're actually making a better world for, unfortunately, like Christian said, just African American people that happen to just be predominantly dealt with having this disease, which is just terrible. And also similar to the CNN article, it also shows how CRISPR works in the editing of these genes, like I said, and to help cure sickle cell anemia. Just a couple of things I'd like to add. just between the two articles. Like the CNN article, like I said, was just more to the general public and patient experience, and is really conveyed a emotional kind of language towards the audience, as the other peer-reviewed article, the NGM article, was like a more medical and scientific article and was more kind of giving information to a more medical and scientific audience, and went, like I said, went way more into gene editing and clinical trial data, and gave a very detailed and technical explanation of that, and gave graphs and tables, and as well as had tons of scientific references and had tons of scientific evidence to back up all the data. And that's all I have to talk about between those two.
Finally, we just have our a few of our references. Obviously, we have our original reference and article, which was the FDA approves the two gene therapies. We have the 5 diseases that could be treated with CRISPR, and then following by the New England Journal, which was the case against gene editing for sickle cell disease. And that's all we have. So thank you.
Thank you.
I'm supposed to click stop sharing. I don't want to mess this up.
I think you just gotta stop the recording. You might have got it, I'm not sure.
I think I did it.
When you go to the more and the record and transcribe tab, does it say to stop for you? Stop recording?
Oh yeah, it does, yeah.
I was gonna click it, but I wasn't sure if that